This content is AI generated for informational purposes only and should not be directly used for clinical decision-making.
Hello and welcome back to the Neurosurgery Hub podcast. I'm Dr. Aris, senior attending in neurosurgery.
And I'm Dr. Alara. Today we're looking at a fascinating new study from the journal Brain Stimulation, published in 2026, titled Stereotactic Rodent-to-Human Approximation of the Mesencephalic Cuneiform Nucleus to Guide Deep Brain Stimulation.
This is a timely piece, Alara. As we see in our clinics, permanent gait dysfunction is unfortunately a very common outcome after severe spinal cord injury. We've historically struggled with how to effectively modulate the recovery of these patients once the initial window of plasticity closes.
Exactly. And the paper really digs into the pathophysiology of why that happens. It attributes this dysfunction to insufficient excitation and control of what they call sublesional spinal locomotor centers. Essentially, the descending input is so disrupted that the spinal cord's own machinery for walking can't be activated.
Right. The engine is there, but the ignition signal from the brain isn't reaching it. That brings us to the mesencephalic locomotor region, or the MLR. For the residents listening, can you remind us what structures make up the MLR according to this study?
Of course. The MLR is a gait control center that consists of the pedunculopontine nucleus, the PPN, which we've discussed before in the context of Parkinson's, and the cuneiform nucleus, or the CNF. What makes this study particularly interesting is its focus on the CNF as a promising approach to restore locomotion.
And the biological rationale is quite sound. Deep brain stimulation of the cuneiform nucleus has been shown to increase reticulospinal drive. We've seen this work quite well in preclinical rodent models of spinal cord injury, where gait was significantly improved.
And that's the bridge this paper is trying to build. Because the MLR is evolutionarily conserved across species, there's a strong argument for translating those rodent successes into human therapy. In fact, the authors point out that early human trials are already underway. They actually cite two specific clinical trial identifiers, NCT 03053791 and NCT 07109804.
It's an exciting frontier, but as a surgeon, my first thought is always about the real estate. The midbrain and brainstem are high-stakes areas. The paper emphasizes that the CNF is in close proximity to several other critical brainstem structures.
That is the crux of the paper's argument for better mapping. Because the CNF is a novel target, empirical human data on the optimal stimulation sites are scarce. If we are going to place DBS electrodes there, the precision has to be absolute to avoid off-target effects in the mesencephalon.
Precisely. In the rodent, we have very clear stereotactic coordinates, but rodent-to-human approximation is a complex task. You aren't just scaling up a brain, you're accounting for different orientations and the density of surrounding nuclei.
And the study notes that for individuals with incomplete lesions, the CNF might be the key to amplifying that remaining reticulospinal drive to a level that can actually trigger the spinal locomotor centers.
It's a shift in how we think about DBS. We've spent decades focusing on the basal ganglia for movement disorders. Now, we're looking further down the brainstem at evolutionarily conserved centers to treat structural connectivity issues like SCI.
It really highlights the importance of this rodent-to-human approximation. Without precise stereotactic guidance, even the best preclinical results won't translate safely to our patients. We need that empirical human data to catch up to the preclinical models.
Well, this study is a significant step in that direction. It provides a framework for how we might localize the cuneiform nucleus more accurately in a clinical setting.
Definitely. It's a great read for anyone interested in the future of functional neurosurgery and spinal cord rehabilitation.
Agreed. That's all the time we have for today. Thank you for joining us on the Neurosurgery Hub. I'm Dr. Aris.
And I'm Dr. Alara. We'll see you in the next episode.